All right, let's jump right in. We're about to go on this amazing molecular detective hunt. When you look at a simple glass of milk, what do you see? Well, to a biochemist, that's not simple at all. It's this incredibly complex, cloudy soup, just jam-packed with thousands of different kinds of proteins. And every single one is like a tiny machine with its own special job. And right there, that's the heart of the problem, isn't it? I mean, a single human cell has tens of thousands of these proteins all working, all bustling around. So let's say you want to figure out what just one of them does. Maybe it's one you think is causing a disease. Well, before you can do anything else, you have to find that one protein and pull it out from that enormous crowd. So where do we even begin? Well, we have to step into the world of what's called the proteome. And honestly, it's like trying to find one specific person in a massive, ever-changing city. Now, the proteome, it's not some static blueprint like the genome. Not at all. Think of it as the actual, active workforce inside a cell, right now. It's totally dynamic. It's constantly changing based on, what kind of cell it is, how old it is, and what's going on around it. Yeah, and this is such a great way to think about the difference. The genome, that's like a massive library with every single book that could ever be written. All the potential is there. But the proteome, that's the specific set of books that are being pulled off the shelf and read this very second. It's all about the action, not just the what if. Okay, so our detective story is underway, but what's the first rule of any investigation? You need a way to spot your suspect. You can't find what you can't see, right? This is where something called an assay comes in, and it's absolutely key. An assay is basically a special test that a scientist creates, and it's designed to do one thing: react only when it finds the unique activity of the specific protein they're hunting for. Maybe it makes the solution change color or absorb light differently. Whatever it is, it's a measurable signal. This assay is the protein's unique fingerprint. It's the thing that lets them look at a sample and go, aha, gotcha, our guy's in here. All right, we've got our fingerprint detector, our assay, all ready to go. So the real work begins. First things first, you got to break open all the cells to let the proteins out. This creates what scientists call a cell soup. And then it's time for the first big, rough sift to get rid of the obvious stuff. And they do this with a process called differential centrifugation. Honestly, you can just think of it like a souped-up, super high-speed spin cycle for molecules. They spin that cell soup faster and faster, and the heavy parts of the cell, like the nucleus, get packed down into a pellet at the bottom first. Then they just test each part with their assay to see which fraction our target protein ended up in. Here's another really clever early-stage trick. It's called salting out. By adding just the right amount of salt, you can make huge groups of proteins suddenly become insoluble. They basically just crash out of the solution, and you can get rid of them. It's a bit of a blunt instrument for sure, but man, is it effective for tossing out a ton of junk right from the get-go. Okay, so the first broad sweep is done. We've narrowed the field. Now it's time to bring in the big guns, the really high-tech forensic tools for some super precise filtering. First up is a technique called gel filtration chromatography. So picture this. You've got a column packed with these tiny porous beads. When you pour your protein mix in, all the little proteins get stuck, wandering through the maze inside the beads. It takes them forever. But the big proteins, they're too big to fit inside, so they just zip right past on the outside and come out the other end first. It's literally a molecular race, and the biggest ones win. Okay, what's next? Well, we can sort them by their electrical charge. This is called ion exchange chromatography. This time, the beads in the column are charged, kind of like tiny magnets. Let's say we use negatively charged beads. Well, any protein with a positive charge is going to stick to them like glue. Everything else, the negative and neutral proteins, just flows right on by. Then, all you have to do is wash the column with some salt water to release your captured protein and collect it. Pretty slick. But this is the real game changer. The most powerful, most precise method of them all is affinity chromatography. This is like designing a custom-made trap for your one specific suspect. Scientists attach a special molecule, the bait, to the beads in the column, and this bait is designed to bind only to the protein they're looking for. It's a perfect lock-and-key fit. Everything else in the whole mixture just washes right through, completely ignored. Then, they just change the conditions to release their protein from the trap. It is, without a doubt, the most brilliant way to fish one single molecule out of a sea of thousands. So, after all that filtering, we're pretty confident we have a pure sample. But you know how it is in science. You can't just think you're right. You have to prove you're right. So it's time for the final lineup, the Protein Identity Parade. And they do this with something called SDS Page. Here's how it works. First, they treat all the proteins with a detergent called SDS. What this does is it unfolds them and coats every single one in a negative charge. Why? Well, this makes it a fair race. It gets rid of any differences in their natural charge or shape. Then, they use an electric field to pull all those negatively charged proteins through a gel. At this point, only one thing matters: size. The little guys can wiggle through the gel's mesh really fast, while the big, bulky ones get tangled up and move much slower. And just look at this. This is the payoff. It's beautiful. Each one of those dark bands you see is a different protein. You are literally looking at the story of our entire hunt. Check out the lane on the far left. That's our starting cell soup. It's just a big, messy smear of hundreds, maybe thousands of proteins. But then, as you look from left to right, with each purification step, you see the bands disappearing, fewer and fewer suspects, until you get to that final lane on the right. One clean, single, strong band. We got 'em. That's our protein. And if the picture wasn't enough, the detective's lab notes, the hard data, tells the exact same story. Just look at that column called specific activity. Remember, that's our assay, our measure of how pure our sample is. Notice how with every single step, that number just skyrockets? At the same time, the total protein column, which is all the other junk, just plummets. It's the story in numbers. And what does that all boil down to? By the time we get to that last step, the purification level is at 3,000. Let that sink in. Our final sample is 3,000 times more pure, more concentrated with our target protein than that messy soup we started with. That is just an incredible feat of molecular sorting. And there's a really good reason for all this painstaking work. There's a saying in biochemistry that really drives everything they do. If you want to get clean, reliable answers about what a protein does, you have to start with a pure sample. You just have to. Any other proteins hanging around, any contaminants, could totally mess up your results and send you down the wrong path. So there you have it. After this incredible multi-step hunt through a molecular haystack, our culprit has been caught. The protein is isolated, but here's the thing. Catching the protein isn't the end of the story. Oh no, it's really just the beginning. Because now that they have this perfectly pure molecule, scientists can finally start asking the real questions, unlocking its secrets, and figuring out its true role in the grand story of life itself.